invite or welcome our instructor, Sue, who will take us through module 5. Thank you, Wanjiro, for clapping for Sue. Let us all clap for Sue. Thank you, Munya. That is great. Welcome, Sue.
Thank you. Thanks, James. Thanks to Maikau. And good to see you all here. Wonderful. And hi to the individuals that are on YouTube. Today, I'm changing things around a bit. I had wanted to play a particular video for you, but I'm going to ask you to watch that in your homework and then to comment on it in the group. And instead, we're going to do a deep dive into bacteria, fungi, and more fungi. We're going to go looking for fungi in our samples based on what we see. Okay, and we might come across some other things, but we're going to be specifically looking for fungi so that we can identify it under the microscope. And I think this is going to be maybe for some of you your first microbial safari. So I wish you all a lot of luck with it. And I'm going to start off first by doing a short presentation. I'll try to zip through the presentation as quickly as I can. Let me just go to sharing screens now so that I can get you clued up on just the importance. We've spoken about it before. We've had other speakers speak about it before. The importance of that bacterial to fungal ratio. What does it mean? We know the fungi are moving nutrients up and down. The nutrients are being produced by everybody else in the cycle. They're also moving water. They're connecting in roots right into the cells in roots, and they're spreading out and giving the plants a much bigger, wider reach. They're also creating the micro and macro aggregates. They're also, doing a whole bunch of other things. Some of it electrical stimulation, some of it, it's just amazing what fungi are doing. All in all, you're going to realize after this that they're so critical, so critical, and our actions on our soils that damage our fungi create a purpose-driven destruction of soils and food and nutrients in our lives. So let me go. We've seen this before. We've seen it many times, right? We just saw it with Dr. Elaine Ingham in the class before when she spoke about the soil food web. And we can see there in the second trophic level, that's where the fungi come in. Extremely important, very early on. We're not waiting for fungi to happen right out on the other side. They're extremely important all the way through the processes. In fact, bacteria and fungi, take those out of the picture and nothing else can survive. We also, very early on in our lesson, we learned that Dr. David Johnson had done this experiment. He explored the differences between yields of crops grown in different mediums and compost, investigating the drivers of the yields. And he found that the levels of nitrogen, phosphorus, potassium, and organic matter didn't contribute logically to increased yields. And what he found was it was that fungal to bacterial ratio. The more fungi, the more bacteria, that ratio in between there, what the balances were like, actually drove yields. We're now going to learn that they actually drive yields across different kinds of plants as well. Why do they drive yields? Okay, there are more nutrients being cycled between the plants and the microbes. There's more aggregation, micro and macro. There's more air, oxygen and nitrogen. There's more moisture. There's water retention. There's more competition, inhibition, and collaboration. There is more toxin remediation and also pest and disease suppression. That's why the yields of those plants were better, because all of those things were happening. So here's a picture, and I'm going to keep overlaying information on it. It talks about the stages of plant succession and the fungal to bacterial ratio. Okay, on the one end, we've got bare ground, and then we've got our pioneer plants, our annuals. Those are plants that can grow on an area that doesn't have a lot of soil. And gradually, as we grow more and more soil, we get perennial plants and grasses, we get shrubs, woody pioneers, short-lived pioneers, and eventually the soils and the systems can manage to withhold and grow old-growth forests. If you look at the bacterial to fungal ratio, what happens there? We go from very bacterial, where we have bare ground, there's not a lot of fungi, all the way up to extremely fungal. Trees need to reach nutrients from so far, whereas if you look down at pioneer plants, they may not need to really. They don't live that long. They don't have woody stems. They don't actually even produce, the carbon materials that are fungal food or fungal built even. So the numbers in the middle there, we've got a 1-to-1 ratio between perennial plants and shrubs. That's where we have, if we're looking under the microscope, we've got a 1-to-1 ratio in terms of biomass of fungi and bacteria. Anything lower than that, it's right down to the 0 on the bare ground, and then higher plants need a much higher fungal to bacterial ratio, and it changes as the plants grow. What we also realize here is that down on that lower end, we have what we call non-mycorrhizal plants. They don't need fungi, okay? There are some plants actually that suppress fungi. You heard about that also with Dr. Elaine Ingham, the brassicas in particular. So your cabbages, your kale, your Brussels sprouts, that entire group of brassicas, they don't need much fungi. They don't need to work with fungi. Some can, but they don't need to. And we find that in our soils and even when we destroy our soils and we just keep pushing nutrients in, we might still be able to grow cabbages. But when you look further up, you look at other plants that need that, along that line as well, you see that they're more woody. You've got little shrubs all the way up to trees. Your maize are going to be down there, and the perennial plants and grasses in that area. They definitely do need mycorrhizae. They need to be colonized. VAM is vesicular arbuscular mycorrhizal fungi. They need it. And if we plant maize or plants that need it in soils that do not produce it, they become weak and subject to pests and diseases. We also spoke about how we had nitrates moving up to ammonium, and that change is happening through the cycling, through the microbes in the soil. As you get to a higher fungal dominance, you get more ammonium. And the plants that cannot manage with ammonium cease to grow. So many of your weedy plants, which don't really need ammonium, they need more than nitrates, they change along that way, okay? So it's important that, when you start moving your soils in the direction of a higher fungal biomass, more successful, healthy plants, especially those that have got woody-ish stems and onwards, fruit trees, grasses, etc., you get less weeds anyway, because the weeds can't survive in soils that predominantly have got an ammonium being produced by the soil food web. If they are, and you've got very weedy conditions underneath your fruit trees, it means that there's something wrong in there, something either wrong with your practices, or some compaction, or chemicals that are being used, or something. But ideally, that should not be the case. When you look at what we grow as humans, our food, we're growing everything down in the brassicas, I said, all the way up to fruit trees. So we need to have some things in a higher fungal dominance. But when we look at soils, in Kenya and many other places, what we find is that our soils are right down there in the highly bacterial, non-mycorrhizal state. Our practices have selected for non-mycorrhizal, highly bacterial soils. Yet we're trying to grow plants and food security depends on us being able to grow these things, but we're doing it in damaged soils because of our actions. Most agricultural soils are here, okay? What we're going to be teaching you is how to make a biocomplete compost that is going to inoculate your soils and move them up in that direction. How are we going to do that? We're going to select the right materials, the right recipes, the right temperatures, so that we can build back the biology, get it into the soil, and move our soils in that direction and change the practices that we need to change in order to keep those microbes fed. And you've heard it time and time again, that liquid carbon cycle. If you do not have living roots and put microbes in the ground, they are just going to die. You're wasting your time. So you need to have living roots all the time, feeding them once we get them in there. And we're slowly going to move our soils in that direction we need to go. So what is biocomplete compost? According to the definition by Elaine Ingham's Soil Food Web School, there are certain recommendations around the biomass of certain organisms. We've spoken how we've got the bacteria and the fungi. We look at a ratio of about 0.3 to 0.8 to 1. Will give you biocomplete, but you want to get it much higher if you're growing, for example, trees. We want to have about 1,000 beneficial protozoa. What are the protozoa? We've spoken about the protozoa being there are some beneficial ones, and there are also some non-beneficials, pathogenic ones. And under the microscope, we can see those. The beneficial protozoas we know are the amoeba, they're the flagellates, and the non-beneficial ones are the ciliates. Ciliates have a very short lifespan. They come in when soils are compact, when soils are flooded. They do, they survive in conditions like that. And then as conditions revert to being more aerobic, they love anaerobic and acidic conditions. As conditions change to become more aerobic and soils begin to get better structure and drain better and filter better, these ciliates leave, they die, or they get consumed, and the good guys come in. We want to have about 100 beneficial nematodes. Now, we've spoken about all the different kinds of nematodes. If any of you had joined the Kenyan discussion the other day on soils, we had somebody, one of the researchers and scientists from ICRAF, who was talking about nematodes as well. And out of these, 20,000 species of nematodes that are in the soil, it's like one of them is damaging to our crops. But if we continue, and those are the root-feeding nematodes, and we hate them, but there are many other nematodes that consume the predatory nematodes. And hopefully I found a beautiful sample today here. I'm going to take us through that to show you the different types of nematodes, and we identify them by their mouth. Those nematodes actually consume the root-feeding nematodes. So we want to have them. We want the predators in there. Remember, it's all about balance. If we think about the Masai Mara or any of our parks, and we think about the numbers of predators in terms of the cats, the lions, the cheetahs, the leopards, and the amount of antelopes. If we had no cats, we would end up with an abundance of antelopes, and we would probably end up grazing places down. There is a balance that comes into play, or we hope, right? And it's the same thing in our soils. We try to select to make that balance. Actinobacteria, those are, again, they look a little bit like under the ground, they look a little bit like fungi, but they're not. They're bacteria that line up in a line, and they form a filamentous kind of strand. So we do call them actinobacteria. They used to be called actinomycetes, which was a word saying that they were fungi, but they're now no longer classified as being fungi. And then omycetes, those are the pathogenic fungi. We don't really want to see them there. Now, there could be some, because you might have pockets of anaerobic stuff, but if you find your population is going over 10% and you've got a bunch of ciliates in there, more than 5 per drop, or more, or even root-feeding nematodes, you know that you've got a problem. So if you're making compost and you've got root-feeding nematodes and things that you're seeing in those materials, you don't really want to use it. Okay, you don't want root-feeding nematodes in your compost, and the reality is there should be, root-feeding nematodes feed off fresh-growing roots, and those should not be in your compost. By the time you're looking at your compost, any that might have come in your plant material should be dead. Okay. Bacteria, we've spoken about beneficial bacteria and pathogenic, and we have both groups for fungi and beneficial and pathogenic fungi have we spoken about. Let's look at the beneficials of both of them. Actually suppress diseases, build soil structure, macro aggregates, retain water. I've gone through this list already. They maintain an alkaline pH, okay? Kenyan soils are acidic. We need these guys, okay? And they exist in an aerobic environment. The disease-causing ones or the pathogenic ones cause disease, release toxins, and they create acidity, and they love anaerobic conditions. So when we make compost, how do we select for beneficial bacteria and fungi? And the way we do it is by understanding what they need. If I was going to invite you all to a party and you were all vegetarian, I'd need to know that, because if the party was to be successful and you don't leave, I would need not to be having a nyama choma party, right? Although I love nyama choma. Anyway, so when we want to invite bacteria and fungi to the party, we need to feed them. What do they eat? So they use enzymes to enable them to consume. I've got writing all over the front of the thing here, but when we look at what bacteria need versus fungi, bacteria need very simple sugars, whereas fungi need more complex nutrients than bacteria, okay? So how do they reproduce? When bacteria accumulate enough, and you can read that, I don't need to read it out to you, they split the cells, we call it binary fission. Some of them, you know, it's really cool to see under the microscope when we're looking at some of those things just splitting, and especially actually when we look at the bigger, the amoeba, very cool to see, we might see some today. Bacteria are the fastest organisms to reproduce, okay? Fungi reproduce in many ways, by spores, buds, by fission, and others, the majority reproduce through spores. When we think about mushrooms, those are the fruiting bodies of fungi. Conditions that they all like are moist, nutritious, simple and complex, and aerobic. So when situation becomes unfavorable, some of them die, some produce spores, or they go into cysts and they can go into dormancy. Some can stay in that status undisturbed for many, many years, and others don't. It all depends on the prevailing conditions, how much energy there was in there, how much food, how much, you know, how long the conditions, how extreme the conditions become in terms of desiccation, dehydration, even heat. But they can quite quickly go into these dormant stages, okay? Or they shut down the metabolic activity and they form those things. Spores can survive, fungal spores, some of them can survive thousands of years. Dr. Laningham was given some spore from one of the Egyptian tombs. It was 4,000-year-old spore, and she managed to germinate it. So it's a good thing, it's a bad thing. We're seeing, as the planet is warming in some areas, we're seeing that there are a lot of, we're hoping they're beneficial fungi, bacteria that are coming and now being you know, released into the atmosphere or surviving. But there could also be some bad guys. So we need to watch out what kind of diseases may have been hidden under all that snow that are now coming to the fore. So when we're looking at compost, our biocomplete composting tips, we will need to develop a good compost recipe. I know a lot of you have got questions. We have to have everything ready before we start. We need to be prepared to document the process through the thermophilic stages. And we need to absolutely adhere to the turns. And I always say get help, have a compost turning and learning party, and be prepared to learn from failed piles, and in general, be very patient. Now, I'm going to stop this here, and I'm going to take you over to a short video that I'm going to show you what fungi look like under the microscope before we go there. It's 20 minutes long, so if you've got any questions, just throw them into the chat. Hi, Manny from Oroville. Throw them into the chat, and I will eventually get to them. After we've watched the video and I'm looking through the microscope, James will take us through answering all of those questions. So I'm now going to share straight on to the video, because it's very important. We need to get that done before we go under the microscope, or you are not going to have a clue at what you're looking at. And when you do, it's so much more fun. Okay, so, oh, sorry, let me just stop that a minute. I just need to do something else here. I need to put the sound on. There we go. Learning about fungi. We'll start by reminding ourselves about the soil food web, which defines for us who eats who in our soils, who's dependent on who, and how collectively all the creatures in the soil stabilize a healthy soil so that we can grow healthy crops, have clean water, and have a healthy overall planet, environment, and healthy us, healthy livestock. The soil food web is divided into trophic levels, with the first being the photosynthesizers, jump to livestock. I'm going to jump. And that what sets fungi apart from other kingdoms of life is chitin, which is a long-chain carbohydrate found in their cell walls. As individual hyphae, that is, individual strands, they're invisible to the naked eye. They're 2 to 10 microns wide, but as they adhere and bundle together, they become the visible mycelia, the structures that you can see on decaying wood. So those are the mycelia. The actual independent strands, the hyphae, these are the ones that we see under the microscope. And these are 2 to 10 microns wide. Our bacteria, in comparison, are only 1 micron wide. So they're significantly bigger than bacteria in terms of width, but of course very, very different shapes. Nicole gives us a good visual reference by further describing the soil fungi as being like the cement and stones that build our homes. They're the vital part of the foundation in the soil structure. They're also, though, the neural network of the soil, sending information across thousands of acres, communicating insect attack, instigating plant defenses, and also sharing resources. Estimates point to at least 70,000 species of soil fungi across the world. These include yeasts, molds, and mushrooms. However, this figure is just an estimate. Many more are unknown. You can see that they're almost like a drip irrigation system under the ground, connecting different plants, bringing different benefits in terms of different resources, connecting plants in a neural way. If you imagine our own nerve system, if somebody bangs your toe, you feel it and pull your foot away. You don't just feel it in your toe, you almost feel it across your body. And this is almost how the underground are connected in terms of their nerves. So you can imagine what happens when we go in there with a djembe or a plow and we dig all of this up. We break the communications, we break the spread of resources, and we actually do more damage to the plants growing around and above the ground than we realize, because we're cutting them off from these fungal networks. Soil fungi groups can be categorized as having 3 major functions in the soil: mutualists, saprophytes, and pathogens.
Hey, I'm Joey, and I'm one of the 30 AI voices from SpeechLab. You listened to this video until now and
The first of this group, the mutualists, offer an array of benefits to plant and animals. There are examples of animals that choose to partner with and even farm fungi. Can you imagine? There are animals that farm. These include leafcutter ants, stingless bees, beetles, and termites. So next time you look at a termite mound, think about this. They cultivate the fungi in a fungus garden, comprising a few hundred fungus combs, structures built from chewed up grass and wood, and inoculated with fungal spores. Much the same way when we try to grow mushrooms, we do the same thing. Each year, these fungi in the termite mounds produce a crop of large mushrooms. In some areas, these mushrooms are known locally as omajoa, which are highly prized as a delicacy. So next time you're looking at these mushrooms around a termite mound, just imagine, like us, there are insects that are farmers. That's just so cool. Lichen and algae require this mutual relationship with many species of fungi for their survival too. If you look on the trees around you, you'll see these little things called lichen. Next time you see them in your environment, take a very close look to see if you can see their fungal support system. Lichen are a complex life form that is a symbiotic partnership of 2 separate organisms, a fungus and an algae. The dominant partner is a fungus, which gives a lichen the majority of its characteristics, from its shape to its fruiting bodies. We have them all around us. Mycorrhizal fungi, micro meaning fungal and rhizal meaning roots, are mutualists which live on and in plant roots, creating a symbiotic relationship between the plant, fungus, and their bacterial partners. Remember, down there in the root zone, in the rhizosphere, are the bacteria we've learned about. And now we're seeing that actually there is a relationship between the bacteria, the fungi, and the plant roots. The mycorrhizae reduce plant stress. They offer root protection and water nutrient uptake in exchange for carbohydrates, carbon, energy, and fatty acids. Remember, we said earlier that what sets fungi apart from other kingdoms of life is chitin, which is long-chain carbohydrate found in their walls. Well, they need to get this somewhere, and they get it from the plants. So they give the plants what the plant needs, and the plant, through photosynthesis, brings down those carbohydrates, carbon, energy, and fatty acids, and allows the fungi to access these so that the fungi can grow. Their hyphae are much finer and can travel much further than plant roots. In fact, plants that have fungi down there in their roots increase the surface area of access to water and nutrient uptake by up to 40 times. So who wouldn't want some in their farming system? Mycorrhizal fungi are a critical and significant component of healthy soil ecosystem, making up as much as 1/3 of the microbial biomass in healthy soils. Over 90% of plant families of 250,000 plant species have this mycorrhizal relationship, supporting healthy, resilient plant growth. So we really, really depend and need these mycorrhizal fungi, this special group of fungi connecting underground. Saprophytic fungi are the largest group of macro fungi responsible for breaking down and recycling dead plant and animal material. These are the fruit bodies you see on dead trees, litter, animal bones, even on feces. Saprophytic fungi release enzymes to break down and digest the lignin, the cellulose, or the chitin in this material into simple soluble compounds that can be absorbed by them and by plants as nutrients. In so doing, they play a vital role in reducing the accumulation of dead organic material and in recycling essential nutrients, particularly carbon and nitrogen. Without these digestive activities, forests would disappear under a mountain of logs and leaves. And then we come to pathogenic fungi. Pathogenic fungi are fungi that cause disease in humans or other organisms. You hear me talking about fungi or fungi, they're the same thing. Approximately 300 fungi are known to be pathogenic to humans. Many more fungi are known to be pathogenic to plant life than those of the animal kingdom. The study of fungi pathogenic to humans is called medical mycology. We've all heard of athlete's foot, candidas, and ringworm. And in plants, we're all familiar with fusarium, mildews, rusts, botrytis. Many of the fungi that we learn about are the pathogenic ones because we try to control these, but we very rarely learn about those that we depend on for symbiotic relationships. When we go and address some of the pathogenic fungi in our agricultural systems, we kill the good fungi as well. The same way when we use pesticides to kill some insects, we kill the beneficial insects too.
An 8-second ear trick for anyone with memory loss. Did you know that memory decline is a completely reversible condition?
Mushrooms are the fruiting bodies of some fungi. So next time you're looking at mushrooms in the shops or in your garden or anywhere, just know that they're the fruiting bodies of some fungi. So let's look at how they reproduce. Most fungi reproduce through asexual reproduction. That means that they don't need a female and a male connection, but rather the young get all of their genes from a single parent. Although fragmentation, that's pieces, fission, that's joining, and budding are methods of asexual reproduction in a number of fungi, the majority reproduce asexually by the formation of spores. Now, where this is very interesting is also looking under the microscope. We can see, when we see fungal spores, we can actually see the mycorrhizal fungal spores. We can actually even see the pathogenic fungal spores, which give us an indication of what is happening in our soils, or what is about to happen, or what can possibly happen in our soils under the conditions that these spores would actually come alive. What we do know as well is that spores can stay in the soil for tens of thousands of years. Here we can see a variety of fungal spores as seen under the microscope. As you can see, they come in a variety of colours and shapes. Some of them there are 10 microns, some of them are 50 microns. So the spores are quite large quite wide compared to the actual fungi that they produce, or the hyphae that they produce. These pictures of spores were taken in our lab. The first is of wild mushrooms that sprouted after the rains, and the second two were in compost and a vermiculture sample. Their large size and dark colour indicates that they're from good soil fungi, and perhaps even mycorrhizal fungi. Fungi are everywhere. Here we can see fungi in a packet of tomato juice. We looked at the green mold on top of the fungi under the microscope and saw that in fact it was green spore. We need these fungi. This indicates to us that this tomato paste is not good enough for us to eat. So fungi are really great in indicating to us that some things might be dangerous for us to eat because they've reached their expiry date and perhaps are even producing toxics now with other organisms that may be in them. So when you see fungi growing on things, we know our automatic reaction is, oops, we need to chuck this out. So look at fungi as being your friends. As we've seen in some of the pictures above, pathogenic fungi are not desirable. When we look at these under the microscope, we can see some very obvious differences. Color, diameter, and cross walls are quick indicators of good or pathogenic fungi. On the left there you can see what we consider to be good or beneficial fungi, and you can see that the color is a brownish color, the diameter is quite wide, we've said from 2 to 10 microns, and we can also see the cross walls. We call these cross walls septa or septae, and sometimes you can also even see oxalic acid crystals on the outsides of the fungal walls. This is where the fungi are changing their we call it the cation exchange, where they're changing their ions or attracting negative or positive ions onto the fungi wall, and in order for the fungi then to absorb those nutrients, using their enzymes and absorb those nutrients into the fungal, into their systems, and then use those to pass on, move on, grow with whatever it is that they want to do with them. On the left side there, you can see the non-desirable soil fungi. So typically they are colorless under the microscope. Their diameter is quite narrow in comparison to good fungi. They are approximately 1.5 to 2.5 microns wide, or to 3.5 in some cases, but typically about 1.5 to 2.5. They may have cross walls or may not. Some of them have periodic or irrationally spaced septa or cross walls, whereas with the good fungi, they're normally, if you look at them, they're the same distance, equidistance apart, but they're not like that most of the time in the bad fungi. When we look through a sample, through our microscopes, and we're looking through your soils or compost, we're looking to identify which fungi are present. If you look closely at this one, you can see a tiny fungal strand right in the middle of this picture. This is only at a 40 magnification, so 40 times larger than it is. But to absolutely identify it, we would need to increase the magnification all the way up to 400x. That's 400 times. And here we can see some good fungi again. the nice brown color, clear and evenly distanced septa, and the picture down at the bottom, you can actually see the little granules of the oxalates on the outside of the fungi. And then here are some pathogenic fungi. They're narrow, they're messy, they're transparent with irregular septa, and they can grow really, really quickly.
What's the first thing you do when you wake up in the morning? If you're checking your phone, stop that. Instead, try this technique from the mind side.
We know that fungi are great decomposers. They therefore hold nutrients locked up in their structure, and they also predate or predated on by other organisms in the soil food web. And these organisms cycle these nutrients back into the soil in plant available forms through the poop loop. So again, looking at this picture above the sun there, we've got living plants that break down into dead organic matter. That dead organic matter is then taken up by the second of the trophic level in the soils, the decomposers, the saprophytic fungi, the aerobic bacteria, and they in turn accumulate nitrogen. And then these are predated on by protozoa, amoeba, flagellates, and ciliates that poop, and others actually, and micro and macroarthropods. But when these excrete their poop, they put back those nutrients that they have got from the bacteria, from the fungi, they put it back into the soil as plant available nutrients. So that's how the poop loop works, and that's how the nutrient cycling works inside the soil. So you can see why it's absolutely beneficial. If you just look at this picture and imagine that you didn't have bacteria or fungi, there would be that gap. There wouldn't be proper nutrient cycling. Or if you didn't have any of those that predated on those, and you had very highly bacterial soils, well, what would happen then? And this next picture shows us that the trophic levels underground influence plant succession above the ground. If we have very, very highly bacterial soils, we're likely only to get very first succession plants, the pioneer plants, those that come in because they can, that don't need to have such a mature and balanced soil ecosystem, and those are the weeds. So when we think about why we have so many weeds in our soils, and with what we know now, disrupting our soils actually creates the best environment for weeds. We break up the fungal pathways so that the maturity of the soil, the maturity of the soil food web, the trophic levels don't build out as they can because they're always disrupted. And we get those pioneers and those weeds. So we're going to be learning how we can move our soils to growing those trophic levels with as little disruption as possible, yet still able to produce food and crops above the soils. We know that most of our farmers and most of us have seen declining crop yields over time with current agricultural practices, and a lot of that is because we've disturbed the soil food web. So we need to learn to grow with the soil food web. So, for your homework next week, take a walk around your farm and look for fungi or mushrooms. Look in the trees, look under wood on the ground, look in your compost pile, look in your animal manures. Look all over. Wherever you see any decomposing material, you're bound to find fungi at work. Take some pictures and post them in your iCal soil biology group. The WhatsApp.
Pictures. Look for mushrooms. Look for fungi. I'm pacing us through this today because I want us to go through 3 samples. There's been a lot of stuff there that sounds like it's repetitious. At the end of it, you're going to know what plant succession is, the fungal to bacterial ratio. You're going to know exactly what fungi do, why they're so important. Whilst I set up the microscope here, and I'm going to take my just the background so that you can see what I'm actually doing. Whilst I set up my microscope, it's nighttime here for any of you that are in anywhere that it isn't. James will take questions. This is my microscope here. I've got a camera on the top. I've got 3 lenses that I use when I look at it. I've got my 40, so I talk about 40, I'll be talking about this objective lens. When I talk about 100, I'll be talking about this objective lens. And when I talk about 400, where we're really zooming in and looking at the samples, it will be the 40 objective lens. But we're going to start on the 4, actually. It's a 4, not a 40. But the eyepiece is a 10, so 4 times 10 is 40. I'm going to take a test tube. I'm going to put some water in it, and then I'm going to take a sample. I've got 3 samples I want to go through today. We're going to start with a Johnson Su compost sample, because that's where I found some really cool fungi. And then what we're going to do is, when you see on the screen, I'd like you then to go into the chat. Tell me what you're seeing from what you have heard. And then the next sample, I'm going to show you some nematodes. I've got a beautiful sample of nematodes and some amoeba. So any questions, put them in the chat. James, you can ask, I will hear, I will answer. You can explain what I'm doing while I'm doing, but I can't do and talk at the same time very much. Okay, off we go.
Great. That was really, really incredible session, Sue. And I know the students here have really enjoyed and have really learned, especially the homework part now that I'll be looking forward to seeing all of us send in the fungi and the mushrooms around our area. It will be an amazing thing, because I know you're not even aware that you are living with organisms around you. Everybody is going about their own business. You don't even know they exist somewhere, but now that you'll be looking for it, you'll find them. They are almost everywhere. So I'm looking for questions. Just put your questions in the chat.
Great thing is, because we're just about to get in the rains in Kenya, next couple of weeks, we're going to see mushrooms popping up everywhere.
Yeah, the participants here will be showing us the edible small mushrooms, or they have different names and different uses. Let's be careful. Yeah, let's be careful when doing that, because I remember last year we had incidents of people eating dangerous mushrooms. So let us be cautious. But we are looking forward to that. Yeah. Okay. So we're shaking the samples to detach the microbes from the soil or from the specimen. So she's counting up to 30 times or 30 seconds. You shake it that way for 30. Yeah. So she's counting to 30. And you saw how gentle she was. You don't need to shake it vigorously, because you will kill the microbes. So you gently shake it and counting for 30 seconds. Then you'll now be able to see the microbes and that you were detaching. Okay, what types of mushroom exists? Okay, that's a question from Wesley Rotich.
There are so many. And if you look back at your notes, I explained when we were going through the notes there, you've got your saprophytic mushrooms, you've got your beneficial fungi, you've got your pathogenic ones. Go back into those notes and have a look at that. So what I've done, Barbara, for your interest, I've put in one, I've got a 1 to 5 dilution. So I've got 1 cubic centimeter of Johnson Su compost. This is very, very, very dark. Maybe I need to let it settle a little bit. But I've got 1 centimeter.
Somebody is asking a question there. The ratios of Johnson Su compost to the milliliters of water.
Oh yeah, I thought I was answering that just now.
Oh yeah, sorry.
That's okay.
Sorry, I didn't hear that.
5 to 1.
Yeah.
On the question of edible mushrooms, there is a lot of a lot of caution you need to take if you're wild harvesting mushrooms. If you don't know how to do it, don't do it. We had an incident, as Jane mentioned last year, where there was an entire family, 4 children died from eating toxic mushrooms. It was dry, it was hot. They were coming home from school. They found some mushrooms in a place called Ganze, and they took them and they cooked them and they ate them, and very sad, they died. We tried really hard through a mushroom WhatsApp group that we have with experts all over South Africa and others. We managed to get doctors in the US to work with the family, to work with the doctors in Kenya. But unfortunately, I mean, 9 people were affected, but we lost 4 children. So I wouldn't go with the fly thing. It does sound funny that, you know, when you get flies around, those are, there is, you know, there's a lot of wisdom in the elderly people who know about mushrooms and mushroom connections, but we just need to be really, really careful. If you want to eat mushrooms, I always say stick to the ones that you get in the supermarkets or the stores for now, unless you have some great wisdom. I once asked on a WhatsApp group, because I put pictures of mushrooms on, it was actually on Facebook, and I said, are these edible? And somebody responded and said, everything is edible at least once, which means that, yeah, but you might die. So please be very, very careful with mushrooms. I'm going to switch now to my camera on the microscope. So my picture is going to go off. I'm going to share screens to that. Here we go. Okay, now you guys are in the camera. Put the microscope on. Now this is a Johnson Sioux sample. I'm going to increase the size over here. Maybe not to there, maybe to 33. Where did it go so big? Maybe there. And I'm going to take the light down. Changing the lens. A little bit closer. That is at 100. I'm going to, right in the middle of the screen, I'm going to take this annotate tool so that I can draw. And I've got a pen. I will draw in Word. There is an amoeba there. There is a little piece of fungi right there, and I'm going to move that into the middle. We'll zoom into the amoeba first because they're not usually like that. I'm going to give you some more light. And I'm going to zoom in closer. Now, right in the center is the amoeba, if you look at it. That there is an amoeba. I need to move something that I've got over there. I need to get rid of this thing. Go to my mouse. Yeah, you like to see my screen. That's an amoeba. I'm going to move another amoeba into the middle. Do you see that other amoeba now? That's what they look like. Not all of them. These are called testate amoebas. The testate amoebas, this is what they look like. That's another one there. These are good. You can see their tummies are full of the tiny, weeny little dots that you're seeing bacteria, those little things that are moving. And the tummies of those things are full of them. Now let's go look for some fungi. Okay. So there's a little piece right there. I brought it right into that central point there. There's a little piece of fungi there. We've seen it. It's brown in color. It's considered good. Let's go see if we see a video of that was there when we do that. There is a much bigger piece, a much darker piece, a good piece. These are all good indicators of that dark piece there. Let me just take away this thing again. good indicators of good fungi. Remember, the darker the better. Let's move around a little bit more. These lovely gold colors in here. Let's see. Again, we've got some more amoebas. Those are amoeba. That's an amoeba. All these guys that are in here. Whoops. To do the nutrient cycling. You want to see a lot more fungi than I'm seeing in here. Just take those off. Those brown things again. Are there any questions coming up in the chat, James?
I don't see, okay. I don't see much nematodes, middle, bottom. Well, I can see people answering. Cannot see much questions here.
That's amoeba going under binary fission. So it's one amoeba becoming two. Here's another beautiful amoeba. And actually, there are a few different ones here if you look closely. All of these, even these little gold guys, that's an amoeba there, that's one there. This is different species, that's another one there. Let me just take the markers off them and give you some light. So the cover slip is about 9 microns thick, the liquid underneath. There's something moving on the side. Let's see what that is. And then within those 9 microns, things move around. Now we're looking right up at the top, at that piece of a bit of an aggregate. You see all the bacteria that are up in that level just popping around. As we're going deeper now down to the bottom, we get to see things that are rested on the bottom of the cover slip. So it seems to be, from a microbe's perspective, it's quite deep. This here is it's like a sausage. It's not 100% sure what it is, but I have a feeling that it is a part of the Salvinia plant that I put in the compost. It takes a long time to break down, but I'm not 100% sure. I'm looking for fungi because you just see one. I have seen a lot in the sample area. This tends to be what happens. There's another piece right there. It tends to be what happens. You look at something, you know exactly what it is. The sample has everything in it, then you come and look at it a few minutes later, and everything changes. Which is why I actually do a prep for a class quite soon before, or with quite a short delay before I show you, because things change rapidly. Okay. This sample in my office, as if it begins to dry down, by morning, the biology will have changed in here. It can be that sensitive. In a big field, it might not be as sensitive, but in a small pot that is drying quite quickly. This funny looking thing here, which looks like somebody's legs in pajamas, is actually part of a trichome, which is a protective part of the plant, which has been invaded by a kind of a fungi. And that's what it looks like as it's being broken down. That part of the plant, this trichome is made of silica, sorry. So it's quite hard for it to break down. The brown thing in the middle there is likely to be a fungal spore. So sometimes we look for spores, but we might see many, but we don't really count them because we have no idea how old they are, whether they are actually going to grow out or not. So when you're doing an assessment, you're looking for fungi that is actually growing. Let me just zoom into the middle of the video somewhere. Big spiky ball thing. Quite interesting. It's got little red dots on it. How cool is that? I don't know what that is. It's like a disco ball. with little red dots on it. Very cool. There's another strand of fungi right there. You're probably wondering,
Yeah, and there is a question there. Somebody is asking how do you count fungal strands? And she's wondering, is this Johnson Su compost?
This is Johnson compost, and guess who's here? Guess who this is? I was just about to say to you, there's not a lot of movement. Look at that. I can tell from his face that that is an omnivore. That's a nematode right there. That nematode would kill root feeder nematodes. It would consume them, would predate on them. See how quickly it's moving? I mean, it's still, we identify it by its mouth parts. Mouth parts of omnivores are like huge big hoovers. You can see in its belly, it's being an omnivore, it also eats bacteria. And if we go down its body here, you'll see that its body is full of bacteria. You can zoom in. See that? It's just been eating away at the bacteria, nutrient cycling food. I'm going to move out so that you can actually see how big this is. I'm moving out to the 100 size screen so you can just see. It's quite a big creature. Do you see the nematode there? James, you can see?
Yes, I can see. I can see. Sorry, I was on mute.
These are good guys. You want them in the soil. Okay. We're going to go in again and look at it to see what it's actually doing. Non-still nematode. You know, I think they bring a lot of life in the soil because everything else moves so slowly. And then you come across these guys and they can just be nipping around and moving so quickly. Okay, we're just going to move on a little bit. There is an egg of something. Yeah, an organism's egg. It could be. I don't think it's it could be actually a nematode from a different kind of maybe there's such a sausage-like shaped eggs, but they're normally quite big. What else have we seen? That's really cool. We saw that. Oh, this is a pretty thing. You see this? You won't believe this, but this thing here, you see this thing in here with the thin lines on it? It's actually a scale of a wing of a creature, a very, very small creature, smaller than probably even a mosquito. And then you have, you'd be surprised at the amount of scales that they have on their wings. What is this thing here? This is interesting. It's a blob of something that's got a few people in it. Again, the aggregates that I've not been able to shape the microorganisms off. Some of them still holding onto the stuff. I'm going to leave you with this screen on while I just set the next slide with the one that's got more nematodes in it, and that is my sample number 4. It's a compost sample. So what can you see in that picture? You can throw the names of the things that you can see into the chat. James, read it out to me. Let's see if
Somebody wrote, I can see nematodes, 2 people, nematodes, and then Vivian can see amoeba.
no nematode there.
Yeah, okay. Vivian can see fungi. Wanjiru can see amoeba. Barbara
Yes.
can see amoeba. There's no nematode. J. and Joshua. Okay, Vivian Bass says nematode.
No, no nematodes. No nematodes in there. Now, I'm hoping the sample that I'm going to show you now is
Yeah, that is the fungal strand, and we have
go ahead, James.
yeah, those people who have mentioned that they can see amoeba, that is
Sorry, I'm now changing the slides. So I'm putting another slide on of a compost. So Johnson Slough is a compost system that technically should give you, it's, you know, is made over a longer period of time. There is a theory that you do get more, more microorganisms in there, especially of the fungi. Now this is a different sample. You can see that there's different material in it. It's quite broken down. The reason why I like this sample is because I saw a lot of beautiful nematodes in here, different types. So, okay, there's one in the middle there. You see it moving around. So have a look at it. That's a nematode in there. It's just moving too quickly for us to really see that when we are doing an assessment, we learn to identify them by the mouth parts we have sorted out. So this one is it's also an omnivore. It's got that wide open mouth, and it would be really good to have in your soil if you have root-feeding nematodes. And we're going to move away from it a little bit and see the types. There is what are we seeing there? Just check in the group what you can see. James, tell me what you can see. Seeing there, guys, what are those things? Oh, look, somebody's come by. That's another. Let's go find it. It's a big one. Woo! And quickly. Okay, what we're going to do is we're going to just come out a little bit because it's a very big nematode, and we're going to watch it from. It's supposed to be further. You see how quick it is? It's so fun. It's like they're so busy. They just don't have time. Chaos. And you'd be surprised, these guys can find each other across the slide quite quickly. And then they fight sometimes. You get them fighting and smacking each other, something like barbs on the end of their tails. They can be quite vicious. James, can you see this properly? I'm trying to keep it in the middle of the screen. Hello, James, are you on mute? I can't hear you in my eyes. Hello?
I don't know, so we can see.
You can see, that's great. Thank you, husband.
Yeah.
What's really interesting about this one is it's a female. I can see that she's got eggs in her body. But she's not going to stop for us to take pictures of her eggs. She's not that keen on paparazzi, but we will use this size. By the way, these big things here, you see this fantastic looking thing? Does anybody know what this is? Guess what this is? There's two there. There's another big one here. Any idea? What are people saying in the chat?
Mitochondria, spore.
No, people keep saying.
We have got eggs.
You'd think it was an egg.
Spore. Yeah.
By the way, this thing right in the middle, this round, beautiful orange thing, this is what we call a, let me give you some light on the very close. Now I'll form it again. This is a Arcella amoeba. Look at the colors. Soil really is gold. That amoeba in there is a different type. It's a testate, but it's called an Arcella. And it also does a lot of nutrient cycling, obviously, like all the testates. So that big circle thing that I showed you, I mean, you guys can put the emojis up of laughing at me for all I care, but I once put that picture on a thing on, I thought I was so clever. I thought I had found the most incredible organism. And I put this on, there's a really fantastic group on Facebook. The Soil Food Web microscopy group are there and everybody's posting stuff. And I went and posted that like a real dunderhead. And you know what it is? It's an air bubble. Nothing of any significance other than showing that I was a real rookie at the time. Look at this beautiful fungi here. Look how much there is. This is what we want to see in our samples. Long strands of fungi. Yeah, different widths, different sizes. There's some here. There's some here, then there's this big long one down here, it branches off here, it branches off here again, and there are quite a few different branches on it. Very cool. That's what we want to see. At this sample, I'm going to go out to 200 again because I just know that in here, there are a few different species of nematodes. I want to show you, whilst I can, what some of them look like. Sorry, I'm just going to, like I see them and you see a bit different. I have to keep you yourself. So when we do an assessment, the first thing that we count, ooh, look at this beauty here. Look at this, she is huge. Look at that nematode. Okay, this is definitely I'm at 100, guys. I'm going to go into 400. Give you some light. This is just her face. Oh my God, she's beautiful. You see that? So, she's being very still. She's not dead. Now, I'm going to point out a few things for you. This here, mouth part deep like this. Oh, she's off. She doesn't like me drawing on top of her. Anyway, that mouth part shows us that she is a bacterial feeder. Okay, she can't bite anything. She doesn't have any teeth. The predatory ones have got a tooth where they bite things. You can see her entire body. She's short. You can see here, that's her reproductive area. There on this, well, actually, that's her where she would excrete as well. So let me show you there. That's a bum right there. But it's also where she would lay her eggs. Isn't she beautiful? You can say, yeah. Love it. We want people like we want people. Let's not organisms like that. That's a nice fun. Let's go see if we see any other ones. That was nice coming across like that. Well, it could have been a him actually. I didn't see any eggs to call it a her. Very, very big one. Let's see what else. Let me just focus a bit. And let's see. There's another Arcella. When we do an assessment, the first things we do is we do exactly what I'm doing now. We go across the entire slide. You have 5 minutes to go across the entire slide the way I'm doing it. And we are looking for nematodes. There's another one in there, a small one. I don't know if you can see it. I can see it. I'm going in at 400 now. I'll give you some light. So there's one in there as well. And its head is hiding under that bit of an aggregate. It is alive, it's moving. We don't count dead ones. We only count live ones and we wait a little bit on them to see and then identify them. Pieces above it is a beautiful piece. It's actually a really good compost sample. There's more diversity in organisms than even in that Johnson soup. This is my compost number 4. I know exactly what went into the making of this compost. And if you look at, you can even see here, inside the fungi, in here, you can see things moving within it. There's some stuff moving on the top, but there's some stuff moving within it. Just to give you an idea of, it acts like like drip irrigation. And then you can also see here a receptor, those joints, these things here. There's a joint there, there's a joint here, there's another one over here. Those are the things that we call receptor. James and Hesbon.
Yes.
Could you please give everybody microphones and let people ask questions while we're doing this? I'm just going to be going across now, just trying to see if we find any more nice nematodes. If there are any questions, guys, your mics are on. Try and control the speaking over each other somehow. We'll have to switch them off. Does anybody know what this beautiful thing here is? Look at that.
Somebody there is saying it is fantastic and grateful for the technology that allows us to see your compost pile life.
Isn't it? And this beautiful thing here is a stellate trichome. Your plants have these on the leaves. They use these with their bacteria to harvest the bacteria and the nitrogen that comes through the plants. They also are able to bring toxins through them to protect themselves from organisms as well. They're really, really cool. It's a stellate trichome. I saw a hand up. Go ahead, speak.
Hi, thank you so much. It's really amazing to be able to see all of this. I suppose my question would be, how would you ultimately analyze this pile of compost? How does it compare to others that you've seen? It sounds like it's very high quality, because we are seeing a lot of nematodes, but also fungal strands. I wonder, how much do you want to be seeing of what? And also a question about how old should a sample be? I know you said something about the longer it sits, oh wow, something else, it will look different like the next day. So I was just wondering how that plays a factor.
So, would I consider this to be a good compost? It is not bad. It's not a bad looking compost. I'm going to actually, while I answer that, I'm going to leave us on some stuff that we can be looking at, because I want to show you a third one. You saw the Johnson Sue one was slightly different. Okay, this guy looks like someone new. I was hoping it was. Sometimes they anchor themselves with their tail, and then they just swish around and literally just shake whatever it is that they're eating off the pieces of stuff that we found. I'm just looking for somewhere to leave you while I quickly put in. I'm going to leave you on that beautiful trichome, because it's such a beautiful thing. I love these things. Okay, so let's see, do we get some good color there? They look like sunflowers, but they're not, and they're tiny, they're microscopic. This is, by the way, a 400X. And these guys are made of glass, which is why they survive unbroken in compost for a long time. In fact, trichomes are even used to do carbon dating, because they can survive fire. So when your plants make trichomes with the silica that they get, when we talk about silica in our plants, and how important silica is for cell wall formation. Silica is also extremely important, not just for cell walls, but also for the defense mechanism of the plants, which are the trichomes. These are all over the leaves, all over the stem parts. When you feel a plant and it feels a little bit hairy, it feels a little bit itchy, it's these guys. And they can bring in toxins from the plant to the surface for anything that's jumping on them that makes them, deter and go away. They're actually really, really amazing. So a good compost versus a bad one, how do we know? So to begin with, we follow protocols where we have a form and a measurement system. So we count the numbers of nematodes. We count all the organisms. We physically count the numbers of nematodes. We then identify which they are, and we've done it from a standard dilution of a system. So from one compost pile, we'll take out samples of the material across the pile. We will then take that sample, mix it, and then from there we will take 1 cc, and we might do this several times on the pile if we think the pile is not standard in any way. It could be that it's a large compost pile that is a windrow, for example. So you might want to test 3 or 4 different areas on it to give yourself an average of what biology is like. This, by the way, is another compost. And you'll see that it all looks different. The material looks different. This is compost number 6. It was different material. It's a different, let me just get the light down. So coming back to how would we be measuring it, we would be looking at counting the nematodes, identifying which groups they are. You don't want to see root feeder nematodes, another nice piece of different looking fungi. We do want to see diversity in different types of nematodes. There is in the middle of the screen right now is a naked amoeba. It looks like it's an amoeba without a shell. So it's not a testate amoeba. This one here is a naked amoeba. And then we would, after we've counted the nematodes, we then start counting. We do 25 checks across the entire slide in a very systematic way, 25 fields of view. So one field of view is 450 microns across. You're seeing on your screen now, probably about 100, maybe 200. So double that size in a big round circle is what we see under the microscope. And we look at 25 different fields of view across the entire slide. And we look deep like this, so we look deep through the aggregates and on the top. And if we come across amoeba, we note it down. If we come across, there's another very big piece of fungi. So there's a very big piece there. See that big bit? So that was must have come from quite a big piece. We will count the amoeba. So if, for example, on this screen here, I'm seeing what I can see 1, 2. I would be really zooming in and out to look. I would count the number of amoeba, and I would write them down. I'd wait for 1 minute on each part, every slide that I'm looking at, to see if things go swimming by, because some things swim. Ciliates will come swishing by really, really quickly, and we will miss them if we move on very quickly. We would also count any flagellates that we saw moving. When it comes to the fungi, we would measure the length. See that right there by that amoeba? This little brown thing that looks like a football. That's a fungal spore, but we don't count those, but it's good to see. We would count the length or note the length of the fungi, the width of the fungi, and the color. Then we have all of that information on a data sheet that we then put into software called SMAP. And basically, we will then get a readout of what the microbial diversity, and biomass is like for each of these independent things that we've looked at. We also do a bacterial account afterwards. Okay, so there is a little flagella moving right in the middle of the screen. Can you see it? A tiny little flagella on it, and the funny little things are like, unlike ciliates, have got a lot of cilia on them. They have one or two little flagella, which are like little whip things on their bodies that you can see it on this if you look really closely. And it's maneuvering its way around like a narwhal with that tiny little flagella. It can't go very straight in one direction because it's only got one, it's like a helicopter with one blade. And that's how we also identify that that is not a ciliate. It is a flagellate because it's bumbling across the system. It's not going at a fast speed, whereas a ciliate zoomed past like a speedboat. They don't bundle. So yeah, does that give you enough information? Any more questions on that, Viv?
Yes, thank you so much. I was also just asking about how long you can go, like keeping a sample before the biology changes so much.
So if you're having samples sent into a lab, you really want to get them in and analyzed as quickly as possible. Okay. And the reason is because they change, and then what you're analyzing, this here, by the way, that beautiful thing in the middle there, it's right in the middle of that thing, is a diatom, a beautiful cylindrical rugby-shaped diatom. I'm going to come off this now, and I'm going to come into the group just so that if there are any questions or anything, I can answer them before we slow down or end this. I'm going to take the slide off. Sorry, guys, we can do this again. I hope you enjoyed that. You can show me with a thumbs up if you enjoyed your first microbe safari. I can stay for hours behind this. So yeah, so if you're getting a sample to a lab, what you want to do is you want to get the sample in a way that it stays aerobic. So you don't want it to be in heat. You don't want the sample to heat up. You don't want to ziplock a bag and have no air in it. You want to have, if you're sending your sample to a lab, you want to have at least 2/3 of whatever that container is with air, and 1/3 with your sample, and then you want to get it to your lab as quickly as possible. When you're looking at soil in particular, you do a different protocol for collecting soil from different places so that you get a good representation. My samples that come into the lab, I bring them in these little pots. My compost is just outside. And I know that because this lab is glass walled and with the heat, by morning, this will be drier on the top. And within a day, this will be dry right through. And it will not be representative of the compost pile that I have outside, which I'm keeping in a cool place, as we should, and I'm keeping it moist at about 40% moisture until I'm going to use it. So I'm going to jump into those questions. I can see Vinay, your hands up. Go ahead.
Hi, Sue.
Hi.
Okay, a quick one. Now that you've shown us the different kind of composts, what are the recipes, exact recipes that we can try?
We're going to be doing that in our next class. I'm going to show you exactly how. So now you recognize how important it is to actually get the right compost, or to make the right compost. So if you're feeding these guys and you're building up these microbes, I will show you that in the next class. It's a lot of information to go into. We do it by really focusing on the target group of organisms and making sure that we get the right quality feed in there for them. Yeah?
Well noted.
Okay. So Barbara, you said how old is a Johnson Sioux sample? I've got 2 samples outside. One is older than the other, but one is about 7 months old and one is about a year old now. From my perspective, when my Johnson Sioux, or where we are on the equator, it could just be our materials and everything. Our compost break down really, really, really quickly. So I think, and I've seen many what I call spent composts. So microbes have eaten and everything, and you eventually get to a point where it becomes quite spent, and the levels of microbes begins to drop. because the food value drops as well. So I think that on the equator, our composts, I've seen they're optimal at about 3 to 5 months in terms of microbial activity. In terms of the secondary metabolites, all of the stuff that's sitting in there that's gone through the poop loop, but is now more of a soluble nutrients, bioavailable nutrients for the plants, anything after that is good. And eventually your compost just gets smaller and smaller and smaller in size. So you do want to get it out and use it. Okay, I can see lots of people were guessing those things. Great, great, wonderful. Can we take me off the spotlight, Hesbon? Let's get pictures of everybody there. And I'm just zooming to the end of these questions to see if there are any missed out. There was a lot that I'd not seen in soil biology, and loving what I'm seeing, amazing living organisms. Thank you for that, Peter. It is just the most beautiful world. Somebody said, will you get a chance to do these practicals? You do need a microscope. If you have a microscope, we are going to be doing a microscopy course in time. We've been asked now several times. So we will do that for people that have microscopes and want a little bit of help and can't make it to the Soil Food Web course. And we'll be talking to them about that as well. Is it possible for you to do such microbial tests for my farm soil? So I typically do not do this as a service. James is setting up the first soil microbial lab in Nandi. Who's in Nandi? I don't know how far you are, Madhuri, from Nandi, but James will be able to do an assessment for you. And we've also built some software which results in SMSs of your microbial diversity and biomass that comes back to you in SMS, whoever it was that sent the sample in. So a really quick, we're trying to provide this as a service to farmers where they can get a quick response in their hands about the biology in their soils or their samples, and then information on what they can do to move it forward. So Nightingale has said, I'm wondering for the desert region like Marsabit, how can we build up our soil so that we can actually grow some foods and trees? I had to work in the sand with compost to build up the soil. You know, it's the same way everywhere else, living roots. Okay, you've got to get your living roots into the ground, and you've got to then recognize that it is a photosynthesis that feeds those microbes. So it might be hot. There are some plants that can withstand heat before others. If you look at what they did in Haller Park at the coast, they went from bare rock to forest, and they did it by carefully selecting plants that could actually work in the harshest of conditions and used those as pioneer plants to create shade and soil for the others that then started coming up underneath them. The very same way we've learned about how fungi and lichens and mosses have done that for everywhere else. So we have to go back to basics. Just planting a tree out there in the hot sun, the tree might not like it, might not have microbial or mycorrhizal support, and might get really stressed. Go ahead, Nightingale, your hand is up.
Thank you for that explanation. And specifically to the dry areas like Marsabit, I'd be very interested in digging a little bit deeper because we are actually working on such. And so I would really appreciate that too. That's a possibility.
Perfect. I want you to go online to the iCow webinars. We've done a webinar with a group in Mexico. And they are doing what they call the agave project. Agave also for license. So agave is of the sisal family, the aloe family. They grow in really, really hot conditions because they're succulents, right? So they've used those to grow, to rebuild soils, and also as animal feed. So please do go to that webinar and have a look at that. And if you like, I can connect you to the team in Mexico and they can show you how to do it. We do have plants that we work with in dry areas that we can use for fodder and we can use to build back our soils. Yeah.
Thank you.
If we, you know, I do understand that we have in Kenya said that some of our agroecological zones, we shouldn't be thinking about planting anything. We should just be thinking about animals. But guess what? Those animals require food. The most cost-effective way of rearing animals is if you have your food there being produced for them rather than buying it from outside. So it can be done. The land is dry. It's true, but it has been done in other places. We just need to learn and experiment. We have the same conditions. We even have the same plants. We just haven't done that experiment yet. So we can, yeah. So goodie for the microscopy course. I'm glad that you guys are interested. What are examples of easy living roots one can use to regenerate land? Is agave available locally? It's everywhere. Sisal is agave. Sisal grows in some of the hottest areas. It's growing in Taveta. It's growing in those places because it doesn't need a lot of water. It actually uses the dew and the leaves capture and create runoff of the dew that they get just with changes in temperature, and they water themselves. And the agave do the same kinds of things. So exactly, you have lots of sisal and kilifi. Go to that. I will actually, James, if you could share the link to the agave, 1 million agave project in the WhatsApp group, guys, do watch it. And we've come to the end of today's session. I hope you guys enjoyed your first microbe safari. I love taking you on it. It is addictive. And yeah, we'll see you all tomorrow. So cameras on, microphones on, and goodbye. See you tomorrow.
Jesse, thank you so much.
Pleasure.
Thank you. Bye.
Bye.
Thank you so much.
Bye.
Bye. Thank you.
Bye. Good night.
Bye, everyone. Good night.
Good night.